welcome to Professor SNAB, where we make A-level biology as simple as a cell. So today we're looking at knowing the ultrastructure of plant cells. So we'll be looking at cell walls, chloroplasts, amyloplasts, vacuole, tonoplasts, plasmodesmata, pits, and middle lamella, and be able to compare it with animal cells. So the ultrastructure, and you should know the definition of this, it refers to the detailed structure of cells as seen using an electron microscope. So the cell wall is located outside the cell surface membrane. It is composed mainly of cellulose microfibrils. It provides mechanical strength and support, maintains cell shape, prevents excessive water uptake in cell lysis, freely permeable, and animal cells do not have cell walls. So the middle lamella is a thin layer found between adjacent plant cell walls. They are composed mainly of calcium pectate, and they cement neighboring cells together and provide structural integrity to tissues. And this is not present in animal cells once again. So now we're looking at pits. Also, just to give you guys a quick tip, I would recommend watching this video alongside the diagram in your textbook so you guys can follow through and associate the different features to the actual diagram. So pits are thin regions in the cell wall where secondary wall is absent. Pits allow movement of water and minerals between adjacent cells. The cell wall is often aligned with pits in neighboring cells, and this feature is once again not present in animal cells. So now to look at plasmodesmata. So these are cytoplasmic channels that pass through cell walls. Plasmodesmata contains strand of endoplasmic reticulum, and this is called the desmotubule, and you should be able to mark this in the diagrams as well. Plasmodesmata enables direct cell-to-cell communication and enables transport of ions, sugars, and signaling molecules, and facilitates symplastic transport. Symplastic transport is the movement of water through the living parts of the plant, like the cytoplasm and the connected vacuoles, in contrast to passing through the cell walls. Animal cells do not have plasmodesmata, but they can communicate through structures called gap junctions. These are specialized intercellular channels that connect the cytoplasm to adjacent animal cells, enabling direct communication. So the vacuoles are large, permanent central structures, and they are filled with cell sap. So cell sap is a mixture of water, ions, sugars, and pigments. Vacuoles maintain turgor pressure and store substances and supports cell expansion. And animal cells may have small temporary vacuoles or none at all, and this varies between the cell types. Tonoplasts have partially permeable membranes surrounding the vacuole, and this controls movement of substances into and out of the vacuole and maintains the ion gradients in the cell. This is once again absent in animal cells since animal cells do not have a large central vacuole. Chloroplasts, these are the site for photosynthesis. This is where photosynthesis takes place. Chloroplasts are surrounded by a double membrane. The internal structures include thylakoids arranged into grana and stroma containing enzymes, DNA, and ribosomes. Chlorophyll absorbs light energy, which is required for photosynthesis, and animal cells do not contain chloroplasts. Amyloplasts are a type of leucoplasts. So these are types of non-pigmented plastids. So pigmented plastids would be your chloroplasts, but these are non-pigmented. And they are found in plant cells that are specialized for storing nutrients. So to look at amyloplasts, they are found in storage tissues such as roots and tubers. So tubers are enlarged underground storage organs in plants, and they act like a modified stem. And a good example of this would be potatoes. Amyloplasts are specialized in the synthesis and storage of starch, and these are once again not present in animal cells. So this video, some of you might have noticed, is actually a re-upload because I wanted to add another slide in because there was some confusion associated with plasmolysis and lysis, which is two terms that are quite easy to confuse, and I wasn't really clear about it in the initial video. So plasmolysis is when the cytoplasm shrinks when placed in a hypertonic solution. So the solute concentration in a hypertonic solution is higher outside. So in this case, the water leaves the cell. And lysis is the bursting of a cell. So it bursts because it is placed in a hypotonic solution. So this is when higher water molecule concentration is outside the cell. So this causes water to enter the cell, and obviously because too much water enters the cell can't handle it and it bursts. So that's the difference between plasmolysis and lysis. And so when you read through your textbook, just remember this definition, so it makes it easier to understand where plasmolysis might be taking place and where lysis is taking place. And also, once again, I would recommend rewatching this video so you know where lysis is taking place and where plasmolysis is taking place. So that is all for Professor SNAB today. Hope you found this video useful. If you have any questions, of course, let me know in the comments and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.